Okay, so the goal of this video is to provide an overview of the features of the Ralstonia Diversity Dashboard. This is the full link to where the interactive dashboard is located on a Shiny Apps server. There's also this URL shortener that will bring you to the same location. So. I have preloaded the dashboard. It does take a minute or two to get going, so just to demonstrate that, it's actually already preloaded. It does usually take at least 30 seconds. It is computing some of the scripts to make the visuals in that time. So give it a little bit of patience to get going. Additionally, after about 10 minutes of the web page being open without being used, it will time out. That is because this is available on the free platform of Shiny Apps, and they charge for that time usage. And so there is a cap, but you can just reload it and ask your question again. Okay, this is the dashboard. The main interactions are located on the left-hand panel here, and it continues to scroll down. Each of these is a drop-down menu that indicates what selection of the data set is being displayed in the body of the dashboard. At the top, there will be these interactive panels that give you information about how much of the data set is being shown on the dashboard after the selections have been adjusted. This is the default view with the full data set shown. So there's almost 10,000 isolates from about 300 articles. Been located in about 108 countries or territories, and has been isolated from 501 different hosts. This will be the cumulative host isolations. This is not the host range of any given strain. As a reminder, Ralstonia strains do tend to have broad patterns of host range, but there are limits to their generalism. Okay. So what we see here is a geographical map that is interactive. So each of these points represents the latitude and longitude of a given isolate of Ralstonia. We have interpreted those latitudes and longitudes based on the text-based location name. So in this case, this is a point that is at the geographical center of Sri Lanka. And this is showing one of the strains that has been isolated from Sri Lanka. When you hover over each dot, it will give other metadata to that isolate, the strain name, phylotype, subphylotype, uh, sequevar, host plant that was isolated from text-based location, and the year. In addition to those hover features, there are some other interactive indications. Each of the dots is color-coded by the phylotype, with any strains that are published without the phylotype information being known. Those will be showing up in gray, yellow for phylotype 1, blue for phylotype 2, 3 for phylotype 3, and 4 for phylotype 4. It is worth noting that when this plot is generated by the code, it first paints on all of the phylotype 1s, or all the unknowns, then the phylotype 1s, then the phylotype 2s, 3s, and 4s. So in any location where there are co-occurring uh, strains, the highest, the most recently painted on one may obscure the ability of you to see which other strains are present. We are working on updating that in a second year of funding on this project, but right now this is definitely the beta version. You can zoom with two different ways, these buttons or the zoom. The zoom buttons are probably a little bit easier. After zooming in, you can then pan around and look at information. That issue of the points overlapping becomes less of a problem with a little bit of zoom because now a lot of them are offset a little bit. So we can see from this picture that South Korea has a lot more phylotype 1s reported than phylotype 4s. We have some notes of caution about the map. These are reports where the strains are isolated. There are definitely cases in the literature where someone has a typo and reports the wrong location. There's also cases where Ralstonia has been reported in a location but has been successfully eradicated. So one of those cases is Sweden. So there was an outbreak in the 1970s, but currently it is believed that the pathogen has been completely eradicated from Sweden. Another thing that we were careful to do is if there was clear information that any strain was isolated from plants that were imported into the location, we noted those in our raw database as imported, and none of the imported plants are displayed on this map with geographic location information. If you use this map, it is worth reading our notes of caution that highlight some of what I just verbally went over. You can reset the axes after you're done, and if there's any that you're interested in exporting to, internal notes or a presentation, you can download images or just take a screenshot. In addition to the map, we have a couple visualizations that show up. We have bar charts that show the abundance of different phylotypes by host species or host family. Currently, these are all displayed in the scientific name, but if you have feedback on whether we should use common names, we'd love to hear your feedback in this Google form, which takes feedback and also, is a place to report bugs that you've noticed. So these stacked bar charts are using the same color coding system. Each of them has a few different buttons that you can use to look at some different comparisons. So you can change it from this axis, which is the total numbers, the count, or you can change it to look at proportion. So in this case, we've now clicked the proportion, and you can see that we can now compare proportionality. Most of the squash isolates here have been isolated, are phylotype 1 isolates, whereas you can see that this banana crossing is a vast majority of phylotype 4 isolates. Another banana species, because they are a little complicated, it is shown here with a lot of phylotype 2s, some phylotype 4s, and the occasional phylotype 1. If you do not want to see the unknown information, you can also redisplay it without the unknowns, which gives you a better chance at being able to assess relative abundances across these hosts. And then over here is the same sort of information, but consolidated at the family level. So once again, we can look at a proportionality or look at it on the count sort of level. Down here are graphs that show the location information. So we have country or location, like territory on the left. So what I mean by territory is for any country that has non-contiguous parts. So in the case of the United States, Puerto Rico, Guam, or Hawaii are not contiguous with the continental United States. So we separated those categorically. And similar situation occurs for many territories in France, where there's many island territories and other global territories that are separated from continental France. So we try to separate those wherever the data allows us to. So we have country and we have continent. Once again, we can compare the isolations reported. look at it proportionality or based on raw counts in different countries, and similarly raw counts for the continent level information. At the bottom, we have a table that is not very useful when it is showing the full data set. Of course, you could click 900 pages. You can change the information, but this table can become more useful when you have subsetted the data set to look at just a smaller section of the data. For example, looking at all the isolations from the country of Guatemala, or looking at all the isolations from the plant ginger. Okay. So that's the content of the dashboard. In order to change it from the default view of all data towards targeted questions, there's different drop downs to the left here. The most useful ones are probably going to be phylotype, potentially sequevar, uh, because there is the pandemic lineage that's also known as race 3 Biovar 2. We make it particularly easy to filter specifically for that lineage. We also have host family, host species, and we pulled out a number of vegetatively propagated hosts that may be vehicles for movement of strains in either latent or symptomatic form, since those are plants that are not seed transmitted. So this is a subset of those 500 species that might be of particular interest for looking at vehicles. We manually created this list. If there's any feedback on which of the host species are particularly interesting to look at, we're happy to update that on the form, so please provide feedback. We also have location information at the continent or country level, as well as whether there are genomes available on NCBI. So to filter for one question in the data set, you open the drop-down menu, and the default, you'll see everything is checked. That means everything is showing up on the dashboard. So if we wanted to say only look at, let's say we only want to look at the unknown phylotype strains. So let's click deselect all, only, and then re-click the one that's interesting. So now it's clicked, it displays that information here. Yes, if you have two things selected, it won't tell you both of them, but it will give you that indication. So you probably want to unclick that if you have a specific thing in mind. And it doesn't automatically generate, you have to click this button here that says filter. So after you click that, the R code runs again, and you get this updated view. That's very interesting. I think I've already discovered a bug here. I've never looked at it this way. I'll be reporting this to our code developer, Vienna. Clearly, this did not work perfectly, but if we want to go for one of the probably more common questions of looking at phylotype 1 and clicking filter, we should see all the yellow dots. That is fascinating that this is not updating. What happens? Okay, it updated after I clicked, so it was potentially a little bit laggy. Okay, so we have that information. It has updated. So looking at phylotype 1, we can see that it does have a global distribution, including locations within the United States. It is worth emphasizing that all of these location information, if it's just reported to the country level, then the coordinates for that sample will be at the center of the location. So this is not an isolation in Oklahoma or Kansas, Nebraska. This is just a United States location. But we see that there's been reported strains in Florida at phylotype 1, as well as Louisiana and North Carolina. Okay. So all of those plots also do update here. These aren't particularly useful just on their own, but they could be exported either as a download or a screenshot, and then compiled against a couple different views if that is relevant. So I'm going to go back to the default. That's so interesting. Okay. And so if you then want to look at a different question, then you would click select all. As I did that, then the phylotype went back to all selected. So this goes back to the default selections. You'll want to click that button before changing what you're doing, just so that you don't accidentally cut an intersection of the data that you did not intend. We also can say, look at different host species. Since this is a very large one, we added a text box that allows you to search. So if we start typing tomato, then there are a few different ones. There are some other common names where tomato gets used. Functionally, cultivated tomato is tomato and cherry tomato, like a persicum and pimpinellifolium. So let's display both of those. So we've got those two host species selected. We will click filter. And here we can see a map view of all of the reported isolations of tomato isolates of Ralstonia. So these plots are a little bit out of proportion now, but they're still here where we have it at the family level, and very few. This is one where you might want to do proportionality, just so that you can at least see the phylotype abundance before going back to just those numbers. Yes, and then the location information has updated here as well. So that's the overall view. I'll make a second video where I talk about some specific emerging strains or strains that I know are of concern or of interest to regulators. So I'll be recording those separately.